So we move now in chapter 21 to one of our last chapters in dealing truly with metabolic fuels. If we think about fuels that we truly use for energy or can use for energy, it's carbohydrates, which are our primary kind of hydrophilic circulating fuel, what our body prefers to run on. We've got triglycerides in our lipid kind of metabolism, and again, great source of energy. 9 calories per gram. It's what nature prefers to store excess energy as. And now we've got proteins. And what's unique with protein metabolism is we really want to think about proteins not as necessarily being consumed as a fuel, but really as a building block. We do need to think about their metabolism though, because whether it's endogenous sources, as in turnover of protein in your body, or exogenous sources, if you just consume too many calories and too many amino acids via protein, we do need to think about how these are broken down. So we'll break this chapter into 3 sections. The first section is going to deal with endogenous protein turnover. So proteins in your body that need to be literally tagged for destruction, how is that done? So we'll talk about the different mechanisms that happens in your body and kind of some unique times when it's necessary. We'll move then into kind of an overview of what we need to think about in terms of the metabolism of proteins. And again, we've got a new element that we need to think about. We've got nitrogen that we need to think about. So we'll kind of bring that into the fold and talk about how we deal with metabolism of nitrogen. After that, again, we'll talk about in our second lecture how we break down these amino acid skeletons. And so there's lots of different pathways that are at play here because there's 20 different amino acids. The good news for you guys is there's very few that we're going to study in great detail. We'll talk about a couple that have interesting disease applications and also keep maybe those disease applications in mind for ones we don't cover as well if it's something that you'd like to consider for your final presentation. The last section, and we'll probably only spend a half a day on this and then move into nucleic acids, is dealing with kind of nitrogen and other places in our body. So we'll talk about things like heme, we'll talk about NO as a vasodilator, and then we'll also talk about just nitrogen fixation in general, and we'll talk about the nitrogen cycle, much like we've talked about the carbon cycle. Okay, so not too much we're going to go through in introducing this section. Again, we're getting now into our third sort of metabolic fuel that we can have, but an important point to kind of highlight here is while we have reserves of carbohydrates and glycogen, reserves of carbohydrates and lipid, we really don't have reserves of proteins, or at least not intentional reserves. That's why your muscles will atrophy if you haven't consumed any calories and you need to ultimately keep your blood glucose level at that critical 5 millimolar. And again, beyond that, we have an elemental problem, no pun intended there, with the idea that we've got nitrogen that we need to consider. So proteins, unlike carbohydrates and lipids, have another element that we need to consider with nitrogen. And again, so we've got not only disposal issues, which we'll address here, but need issues, which we'll deal with in a future lecture. So kind of thinking about disposal issues, how we handle that. If we think about the typical structure for an amino acid, we can literally think about breaking apart the amino group from the alpha keto acid skeleton group. And then that alpha keto acid skeleton group, being very carbohydrate-like in terms of its elemental composition, can be broken down by conversion into citric acid cycle intermediates. And then this amino group, again, we'll talk about how we dispose of this. We don't have toxic ammonia that we produce in our bodies, but we use that nitrogen to generate urea. So we'll talk about the urea cycle in this chapter. But importantly, and we'll get to this in our very last section in this chapter, we do need nitrogen. We do not have the ability to fix nitrogen from the atmosphere like we have the ability, or we don't have the ability, but plants have the ability to fix carbon in terms of carbon dioxide. Most plants actually don't have the ability to fix nitrogen. So we'll talk about how that's handled kind of in a global sense in terms of nitrogen fixation. And like I said, in the very end here, we'll talk about other sources of nitrogen in your body. We'll talk about heme degradation, some really interesting things with why bruises go through their different colors. We'll talk about nitric oxide, and again, an important physiological vasodilator. So we'll talk about important things like Viagra. So big picture here, we've got endogenous sources of proteins that we need to think about degrading. And this is largely what the protein degradation that we're considering when we talk about cellular breakdown of proteins. Most of it comes from endogenous sources. A lot of the protein that you consume in your diet is actually used to rebuild proteins. But there's a lot of turnover, whether it's enzymes, whether it's muscle, lots of protein turnover in your body. And again, we can think of this as a giant substrate cycle between proteins and amino acids. And again, we can store nutrients as proteins. So any structural or catalytic proteins that are in your bodies, we can think of those as being reserves of amino acids should we need them. Again, but if any proteins are malfunctioning, we need to have some way of getting rid of them. And even though we've spent most of our time talking about the activation and controlling the activity of proteins as a way to control whether or not they're doing their biochemistry, we need to think about also the availability. So in our cholesterol kind of metabolism, we talked about transcriptional regulation of that protein to kind of control its availability. We also can think about the back end of things and controlling the degradation of proteins as a way of controlling availability. So 3 ways that we'll kind of talk about degrading proteins, and they kind of start with the least specific and kind of get to most specific. So the lysosome, again, we've already talked about the lysosome a little bit in our receptor-mediated endocytosis of cholesterol. You can think of the lysosome as sort of being a stomach of the cell or a garbage disposal. The stomach of the cell is nice because it kind of reminds you about kind of the breakdown that's going to happen there, a lot of hydrolytic activities that are going to happen there. Again, we're going to see that things will kind of be non-specifically degraded there. But again, we can have selective degradation in terms of what might be tagged to find its way to a lysosome. And we call this a KFERQ tag. Again, might not be surprised to see that those are the amino acids. And that allows us to selectively atrophy tissues such as the kidney and the liver. And that's kind of important if you are in a state where you're fasting. And you don't want to atrophy your muscles because that's going to prevent you from getting to food. Other interesting kind of aspects of this are thinking about uterine regression after childbirth. In just about a week and a half, your uterus goes from about 2 kilograms in weight, which is just about under 5 pounds, to about 50 grams. So significant change in the mass of that organ, and that all happens through this degradation pathway. Same thing with rheumatoid arthritis and autoimmune disorder, kind of resulting from the release of lysosomal enzymes into surrounding tissues. An alternative way, still kind of a way that we tag things, just like you might have that KFERQ tag. If you put ubiquitin, which is a small protein, if you tag a protein with ubiquitin, that can essentially be thought of as a death tag. So proteins that are tagged with ubiquitin are going to be tagged for destruction. We'll talk about how they get destroyed in just a minute, but how do you get a death tag? Basically, your N-terminal residue is going to determine whether or not you get one of these tags. Or, for example, being what we call a pest protein. Again, pests referring to being rich in those 4 amino acids. And again, having those phosphorylation sites make these very useful sites for ubiquitination. So ubiquitin is one of these really important proteins that's thought to have both housekeeping and regulatory functions. If you haven't heard of these terms before, something that's considered to be a housekeeping gene just kind of keeps things clean in their proper functioning state, proper balance in the cell. And then regulatory functions allow us to kind of control whether things are on or off. And again, it might have a developmental, it might have a timing issue to it, but those are just 2 terms I wanted to introduce to make sure that they were familiar to you. So once a protein has been tagged with ubiquitin, it finds its way to this large multi-protein complex called the proteasome. We're not really going to look at the structure much of this, but basically you can think of this as just kind of a giant, again, garbage disposal, again, where things go inside of it and then they're selectively hydrolyzed. Why I say selective is it's interesting that the ubiquitin protein itself, that tag, is not degraded, which allows it to be recycled for future use. Okay, so kind of wrapping up with an overview of what we're going to talk about with the rest of this chapter is amino acids kind of have an interesting problem of having this extra element in terms of nitrogen. So again, amino acids are going to be broken down into their nitrogen component, which we are going to dispose of in the urea cycle, and then the carbon skeleton, which can be converted to a variety of different metabolic precursor or metabolic intermediates that have a variety of functions, and we'll talk about those as they come up. But the big picture with amino acids and proteins is the intent is that they're either broken down to the subsequent amino acids to be rebuilt into other proteins, especially those amino acids that are essential, or again, if we don't need them anymore, if we have proteins that need to be turned over, if we've consumed too many proteins, if we have excess amino acids, we need to metabolize them. And so again, this process is going to facilitate that. So what to do with N has a lot to do with whether you are a bird, a fish, or a mammal. For example, ammonia is secreted by fish, uric acid is secreted by a lot of reptiles, and again, we metabolize our nitrogen by the production of urea. So big picture thing here, this is the last slide for our introduction here, is to think about this process of making nitrogen into something that's metabolizable and excretable. So we're going to take amino acids and ultimately make them into two important amino acids. And you want to remember glutamic acid and aspartic acid. Those are two amino acids. We have some chemistry that allows us to funnel all 20 amino acids to either being glutamic acid or aspartic acid. Glutamic acid undergoes what we call oxidative deamination to get rid of its nitrogen, and that ammonium ion is going to become part of urea. So here's the structure for urea. Aspartic acid supplies another nitrogen via the urea cycle to add another nitrogen to the urea molecule. And then the carbon actually comes from bicarbonate. So we have three essential sources for these three main components of urea. We have two nitrogen sources, an aspartic acid and glutamic acid, and then the carbonyl comes from our bicarbonate. So we'll get to some of these details in our second lecture, but I told you that all 20 amino acids are essentially funneled down into two different amino acids, and we do that with transamination chemistry. So again, if we've got alpha-ketoglutarate, that is the alpha-keto acid skeleton of glutamic acid, and the same with oxaloacetate and aspartic acid. So that's why it was important to learn those and remember those from our citric acid chapter. So again, aspartate is going to provide a nitrogen for the urea cycle. Oxidative deamination of glutamate is going to provide an ammonium ion and another nitrogen for urea. And then bicarbonate is going to provide our carbonyl here. So we'll get into those details when we study the amino acid or the urea cycle, but that's all that we have right now for our introduction to amino acid metabolism.